who will most likely respond well to treatment and in many circumstances not relapse. On the other hand, you have adverse. So these are people that will, on the bottom, you can see the red line. These are people that will most likely respond poorly to treatment and in many cases need more experimental treatment or bone marrow transplantation. The two lines in the middle, the yellow and black, represent the intermediate categories, either intermediate 1 and intermediate 2. Both these categories are for people that don't have these stable biomarkers or things in their cytogenetics that put them apart and put them on a more favorable or adverse side. They're the more gray area and they're a lot more difficult to treat. So these 4 categories are based on 2 separate categories: cytogenetics, like I said before, the karyotype, the chromosome makeup, and molecular biomarkers, which we will continue to talk to you about. 2 examples are Fhcr and NPM1 that have clinical applications.
So what are prognostic biomarkers? Biomarkers are measurable substances in an organism which, when present, is indicative of some sort of disease, infection, or environmental exposure. So biomarkers can be found in various levels, including DNA, RNA, and protein levels. So this cartoon kind of shows that new technology such as next generation sequencing has allowed us to sequence the human genome, and that because the human genome is so large and there's over 3 billion pieces, that each base pair is very unique, and if there was a mutation that would rise, then it could result in possible mutations. And so our lab, we're specifically working on the DNA level of biomarkers in which we're detecting mutations such as substitutions, insertions, and deletions, which may lead to be possible biomarkers. So our goal of the Fhcr lab was to detect insertions and deletions. So this visual kind of shows the original sequence as compared to the sequence with the insertion. So you can see right here, this is a single base insertion which causes a frame shift in the original sequence, and it causes the coding sequence to be read differently. And so these insertions are kind of what we're looking for. And during our project this summer, we were assigned 2 genes called RUNX1 and WT1, in which we were assigned to screen patient samples and detect possible insertions or deletions that may be in these 2 genes.
So previously in our lab members have found these insertions, like Elizabeth was talking about. As you can see on this, we have a technique called next-gen sequencing. So this is where our lab found these initial insertions. So that red, like those little eyes, represent that insertion within the gene. The part below, it represents a patient without AML, so without that insertion. Like I said, the one above is a patient with AML. And then likewise, in the next, we have the RUNX1, which was Elizabeth's. That shows the insertion as well. And then the bar above, that's a wild type, so a patient without AML as well.
So in order to do our project, we had to learn a lab technique called fragment analysis, and it consisted of PCR, gel electrophoresis, and gene mapping. So PCR is polymerase chain reaction, in which we are amplifying large amounts of DNA of our patient samples in order to test for our fragment analysis. And so we would, after amplifying our samples in the PCR machine, we would take these samples down and run them through an agro-strill, in which we would separate out and identify and confirm that the amplification did indeed happen. And so as you can see right here, the bright bands indicate that our PCR products did amplify and they're present. The wall on the very left is our DNA ladder, which indicates the size of the ladder. The size of the base pairs of the PCR products. And on the right is the well for water, which is our negative control, in which we want it to be, so if there was any bands present, it would indicate that there was contamination, but in this case, it's not contaminated. So we can move on to the next step, which is diluting our samples and sending them downstairs for gene mapping. So once you send them downstairs, we would get back these results, which show that, so the peaks represent if there was 2 peaks, then it would indicate that a mutation did occur and there was an insertion or deletion that happened. And if there was only 1 peak, then it would represent a wild type, which means there was no insertion or mutation. So in our results, there are 2, these 2 both have double peaks, which indicates they're mutants. And they're mutants for the WT1 and RUNX1 genes.
And also, the reason why we're doing this fragment analysis in relationship to what's already been done in our lab is because that software, that technique, has some discrepancies in its ability to read larger mutations. So although it shows these insertions, we need to complete fragment analysis to make sure that we're weeding out anything that may have been missed and getting these larger mutations that may not have been able to show up on the technique.
And so throughout the summer, what we learned was since PCR was a vital role in our whole process, we learned the concept of dirty room and clean room. And dirty room is where we work with our PCR samples and where all our PCR products were handled. And the clean room is where we prepare for our PCR. So we have to kind of understand the concept of the dirty room and the clean room and really know that we want to avoid contamination as much as possible. So we had to be very careful of always wearing our lab gear and not randomly touching equipment in the lab.
Yeah, and then that was a big thing, controls and contamination. But then along with that, we also learned a lot about the way mutations work and how a single nucleotide insertion, although seeming very small scale and minute, it can have large repercussions and large impact on the body as a whole, often cancer or just impacting this way that the signal pathways are interjected. And then with that, we also learned the importance of biomarkers. So coming in, I had no idea what a biomarker was. I thought it was like a tag that made a cell like shine, but really what it is it allows patients to be better prognosed and diagnosed, and it allows their cancer treatment to be more specialized for who they are, and this can really impact the way a patient is treated and their cancer is fought off. So with that, we want to thank everybody, but primarily we want to thank Dr. Bev, Liza, and Lori Blake, because without you guys, we would be like at home on our couches and it's sad. We also want to thank our PI, Derek Starwell, because he let us into our lab, and our mentor, Era, for being so patient with us despite like 25 contaminations.
And we also want to thank Andrea, Brooke, and Nina as members of the lab for guiding us through the whole process, as well as our program sponsors and the wonderful interns that made this summer really unforgettable and awesome.
Thank you.
Other questions for these ladies? So do you have a bio? There is one?
Yeah, Dan.
Yes, so for the possible outcomes, the intermediates and the adverse and favorable, which of those is the most commonly occurring?
That's a really great question. From my knowledge, I believe it would be the intermediates, because although these biomarkers are common, they're not, like, for sure, they're not staples of leukemia. So like I asked before, that you can have these biomarkers and not have the disease, but what I think is the intermediates, because most people come in and when they're diagnosed, it's like, okay, well, let's see how this works. Like, but my mentor might have a better answer.
There's actually numbers for how many patients are represented by each category. They just had to cover them up with a better readable label.
Oh, yeah.
So it is a intermediate, probably comprising a larger group of patients.